it's actually in some ways easier to start with the conventional ideas of what RNA was about, because it was a very simple, straight, linear, long molecule that was just easily made from a piece of DNA, and it just sort of floated out of the nucleus into the cytoplasm and was then used sort of like a Xerox copy of the DNA to make a protein. So it was a messenger, essentially, and it didn't really matter what its structure was, or the sequence was, everyone felt they understood it very clearly. It's a triplicate code where 3 nucleic acids would define what kind of amino acid for the protein would get put into place. So that was all very terrific and fascinating, and it answered a lot of questions about how you could keep so much information about how to make proteins and get it translated from your library of DNA. And that's what's been the dogma and what's in place and what is still true to this day. And what has become more clear, especially since a couple of guys named Fire and Mello back in, it's almost 20 years ago now, but they discovered that the RNA itself could be like a tiny little messenger, like a little tiny guide strand, and could prime proteins, could actually be taken up by a protein and used to guide that protein to different places around the cell. And that allowed a system to form where you could basically give a protein sort of a zip code that would give it a very specific destination. It would know where to go in the various regions of the cytoplasm, but it could also bring a protein in from the cytoplasm into the nucleus. So the specificity of that just dazzled everybody. To be able to recruit a protein so precisely to just one little, in fact what it does is it uses a little teeny piece of RNA to match up with its complementary piece of RNA. Did I say RNA or DNA? I forgot. But a little piece of RNA to match up with a very long piece of RNA. So if you have RNA floating in sort of the nucleoplasm or in the cytoplasm, you could still find it. So even in, what must be this dense fishbowl of proteins and strands of things and pieces of DNA all sort of floating around, it can hone in very, very precisely to a particular place. And in the nucleus, it can hone right in to the region of DNA that's currently being turned on and is making RNA.
Is that different than the conventional understanding? Or is it over and beyond the conventional?
It's above and beyond now what say a piece of RNA can do. So not only can it be a triplicate code, but it can actually also become a guide strand. And tiny little fragments can be very, very precise guides to these very ancient proteins. So there's been work before that, of course, in the idea that the structure of RNA itself could also be important. So RNA, like DNA forms a nice double helix. But RNA, using the same matching C to G, A to U in the case of RNA, but it's the same nucleic matching rules, will form not only helices, but can form these long stems and then loops. And like these sort of mutant lollipops can form these very exotic shapes. And that it was becoming clear that shape also is very important for chaperoning around different proteins or bringing different areas of, one area far away from a piece of DNA in one place. Maybe it was also helping pull together two different places of DNA to bring things into sort of the same location. So it was clear that secondary structure was also very important, that the RNA is not just this nice floating piece of spaghetti, but it's actually forming these exotic shapes, three-dimensional shapes. And it's tremendously important also to have a certain shape, because it's like a lock and a key. Things will fit together and in a much more precise way. And interestingly, again with RNA, that shape that could lock into one protein under certain conditions may shift and change, and because of temperature or a change in the ions floating around it, or the pH, it can change shapes and be like switched from one form to another.
To a specific advantage, or just by mutation and freak accident?
No, to specific advantage. That there's probably plenty of trial and error and freak experiments done over the millennia of time with this, but the ability to make a structure and then shift it like a switch and get a different series of reactions going, trigger a different cascade just because the temperature had changed, is quite impressive. Proteins do that too, to a certain extent. But the beauty of RNA is that it's not only is the code, the triplicate code of the amino acids that will build the protein, but the code that is also in your DNA is for a shape. DNA is giving you the sequence that you need to make a specific RNA shape, and that shape triggers a series of reactions all on its own, which is really very cool. And opens up a whole new world of possibilities. So not only do you have shapes that proteins can form, but now you have this whole new realm of shapes that the cell can use, these molecules to link things together in all kinds of new ways. So you have the shape that's important, and then you also have the sequence that's important. And again, when you have astonishingly, you can have these very, very tiny little fragments that have a certain sequence that can be these little guides for these very ancient proteins called argonauts. And that's sort of what the story of my film is about. How, what could possibly happen if you had a system that was that precise and was able to target things to one area of DNA to another, or one gene that's active to another gene that's active, and give them a way that they can correspond with each other, basically very directly, only using that argonaut protein to be like the airplane that carries the signal from one gene to the next. There's a whole process by which that happens, but basically that's new stuff, and so it's very exciting, I think.